بسم الله الرحمن الرحيم. إخواني وزملائي أطباء المستقبل إن شاء الله، سعيدة بتواجدي معاكم على قنوات النيل التخصصية ومحاضرات على الهواء. دكتورة حمدية عزت إن شاء الله بعمل معاكم كورس الهيماتولوجي أمراض الدم. وكورس أمراض الدم إحنا بناخده في البكالوريوس طب في مادتي البطنة بفرعها بطنة عامة أمراض دم أو الفرع التخصصي وهو التحاليل الطبية. هنبتدي محاضرة النهاردة إن شاء الله عن الهيماتوبييسيس. بحب أقول شوية كلمات بالعربي. لو في الجامعة ما بقولش كلمات بالعربي، لكن هنا إن ساعات في أقاربنا ومعارفنا وأهالينا ساعات بيحبوا يتفرجوا من باب الاستمتاع بالمعلومة. يعني إيه هيماتوبييسيس؟ Hematopoiesis is the formation of blood or all cell type of the blood in the living body. يبقى أنا بتكلم على الهيماتوبييسيس اللي هو formation of all blood cell component in the human body. Before we will discuss how hematopoiesis occur, we have first to know what are the function of the blood. The blood is the mean of transportation of substance throughout the body. The blood distributes nutrient, oxygen, hormones, antibody, and it have a cell specialized in defense mechanism, and it collects the residual of and the nitrogen waste and carbon dioxide from the body to go out. Okay, so we need the blood to perform our metabolites, our living, our lifestyles. All the cell in the body is need a blood. Okay, so what are the constituents of the blood? Blood is made of liquid and cellular part. The fluid part is called plasma, and in this plasma there are several protein and substances like protein, lipids, carbohydrate, and mineral salts. Cellular constituents of the blood are known as blood corpuscles. These blood corpuscles have three compartments comprised of erythrocyte, which we call red blood cells, كرات الدم الحمراء, and leukocytes, which is known as white blood cells, كرات الدم البيضاء, and platelet, الصفائح الدموية. يبقى احنا عندنا البلاد عبارة عن cellular compartment و liquid compartment. Liquid, which is a plasma and its suspended proteins and minerals and salts inside this plasma, and the cellular compartment, which is erythrocytes, leukocytes, and platelet. What are the main constituents of the plasma? Plasma contains proteins, albumin, globulin, alpha 1, alpha 2, B and beta globulin. It contains fibrinogen. It contains clotting factors, which are important in stop bleeding and to maintain the blood to be fluid within the vasculature. It also contains amino acids needed for protein content, and it have a peptide, it have glucose, lipid, lactic acid, and have a variety of hormones and vitamins, and contain uric acid, blood urea, nitrogen, carbon dioxide, etc. So plasma contain a lot of substance. Okay. So again, whole blood are formed of fluid, plasma, cell, white blood corpuscles, platelet, RBCs. We have to know what are the mean by in the physiology. Maybe in the year 2, number 2 in the college, you take the what about the clotting pathway in the physiology department. I know that we have clotted blood in the sample. The clotted blood, it's a serum which it doesn't contain fibrinogen, and the plasma which is contain fibrinogen. Fibrinogen is an important coagulation factors that maintain the body in the fluid state. If this fibrinogen is converted into fibrin, that means that the clot is formed. We have to put anticoagulant for the blood sample to have plasma. If we don't put a blood an anticoagulant, that means that I will get serum from the patient. Okay, this is to know if you send to the lab, it will need a serum sample or plasma sample. This is the difference between the serum sample and the plasma sample. Plasma sample contain anticoagulant and serum sample does not contain anticoagulant. We leave the blood to be clotted. The serum sample doesn't contain fibrinogen and the plasma sample contain fibrinogen. Here is the plasma. This is the blood cells, red blood cells, and then white blood cells and platelet. So hematopoiesis is come from the Greek word to make blood formation. Sometimes in the book you read it is hematopoiesis or hemopoiesis. So blood formation or formation of blood component. Okay. What are the cells or who are responsible for this hematopoiesis process? The primitive cells is called hematopoietic stem cells. Where are these cells? These cells present are in the marrow, in the bone marrow, and it gives rise to mature blood cells and differentiate blood cells, different blood cells, after several kind of division, and then proliferation, differentiation, and maturation. So, what are the character of this stem cell? Stem cell also, the other name you may read in other textbook is called or referred to pluripotent cells. Pluripotent cells. What's the meaning of pluripotent? Pluripotent, it means that it can give rise to different kind of cells, a variety of cells. Okay, there are often considered to these stem cells considered to as a building block of the life because they are essential for life. Okay, each pluripotent cell function to form either new stem cell or precursor cell that will eventually form a specific type of the blood cells. So during cell division, this stem cells is divided into one stem cells to keep the ability to maintain the self renewal. What is the meaning of self renewal? To maintain the stem cell pool in the bone marrow, and the other daughter cells is divided and mature into well differentiated cell. Okay. So again, hematopoiesis is a product or production of blood or formation of blood. Where are the hematopoietic system? Where it is located in the bone marrow, spleen, liver, thymus, lymph node. This is the hematopoietic organs. This is the bone marrow biopsy. Okay. This picture shows bone marrow. The bone marrow is formed of bone trabeculae, okay, and fat cells, and all of these are hematopoietic cells. Okay. The amount of fat cell in relation to hematopoietic cells is different and vary according to the age. In young infant and child, most of the marrow are red marrow. This means it's active marrow. With older age, the red marrow is replaced by fatty marrow, and the number of the fat cell and the percentage of the fat cell start to increase, and the amount of the hematopoietic cell start to decrease. So the bone marrow is essential for all the bone produce red cell until person is around the 5 years old. Where are the location of this bone marrow exactly? Tibia, femur, and vertebra, sternum, pelvis, ribs, and cranial bone. Okay. Is this from the start? No. What about during development, intrauterine life? Okay. Of course, all of you're not going to the college from the start. No, you have to pass to KG1, KG2, and then to the school, and after that you come to medical college. So this is how the blood is also come. There is a stage of development intrauterine and after birth. So during intrauterine life, hematopoietic or hematopoiesis take place in the yolk sac and then in the liver and the spleen. The liver and the spleen are important site of hematopoiesis during fetal life. But after birth, the main site of production of hematopoietic element is in the bone marrow. Specifically, the flat bone, as I mentioned before, flat bone like vertebrae, lips, ribs, scapula, and sternum and hips. Hematopoiesis occur in the bone marrow where erythrocyte, this is the blood cells that I found in the peripheral smear, erythrocyte, leukocytes, and the platelet are made. And in the lymphoid tissue where the lymphocytes, the B and T lymphocytes are mature and formed. The liver and the spleen, as I mentioned before, is the main site of intrauterine or hematopoiesis during fetal life, intrauterine. But after birth, if there is a special condition, this can resume their original function by a process called extramedullary hematopoiesis due to increased demand, like in case of severe hemolysis. We'll talk later about conditions called hemoglobinopathy, such as thalassemia and sickle cell anemia, in which there is hepatosplenomegaly due to increased RBC destruction, and the liver and the spleen want to replace this loss of RBCs and resume their original function and perform RBCs. So as I mentioned, hematopoietic phase, embryonic phase or prenatal, mesodermal, which is in the yolk sac, then hepatic, and then myeloid. Myeloid period, which is referred to the bone marrow. Postnatal, medullary hematopoiesis, the bone marrow, specifically in the flat bone, and extramedullary hematopoiesis in special condition where there is increased demand and hemolysis. So during 10th week of the gestation till the entire second trimester, liver and the spleen are the major sites of hematopoiesis. Proliferation of early hematopoietic cells take place, then differentiation of hematopoietic precursors. Precursor means that the primitive cells, the cells that will be mature, it is the stage before the differentiated stage that we see in the peripheral smear. So we don't see the precursor normally in the peripheral smear. Okay. The third trimester during intrauterine life, there is a shift to medullary cavity of the bone, and as I mentioned, the main site of hematopoiesis is the long bone and the shaft of long bone and the flat bone in the vertebrae, lips, sternum, scapula, and skull. This is a nice diagram show the hematopoiesis during intrauterine life, and then it starts in the yolk sac, then the liver and the spleen take place. Then by the end of the intrauterine, from the start of the 4th month of gestation, the bone marrow takes the upper hand of hematopoiesis and continues throughout life. So after birth, the main site of hematopoiesis is the bone marrow. This is a nice picture of bone marrow biopsy. This is the fat cell and this is the hematopoietic cells. Okay. What are these hematopoietic stem cells? I said this is the blood cells that give rise to all the blood cell components. They are located where? In the red bone marrow, which are contained in the core of most bone. Stem cells are undifferentiated cells that are able to differentiate into other type of specialized cells. It can give us to platelet, white blood cells, and erythrocytes. The stem cell of bone marrow originate the differentiated blood cells. So we said that it is the mother cells It is the primitive cells of all blood elements. According to the stimulus from a specific growth factor, the stem cells are turned into red blood cells, leukocytes, or megakaryocytes. So the stem cells have to be committed and exposed to specialized growth factor to differentiate into and to proliferate first, and then differentiate and mature into a specific cell type. That means that we have different growth factors. Like if the patient have hemorrhage and he want to replace, he will not replace the white pieces count. No, he will replace the erythrocytes. So which cell he want to, the stem cells will need to perform? It will need to perform erythrocytes. So it needs the erythropoietin, okay, to stimulate erythropoiesis. Okay. If there is hemorrhage, so I need to stop bleeding. So these stem cells are committed through a specific growth factor called thrombopoietin to form the megakaryocytes, which give rise to the platelet, which aids in the primary hemostatic plug to seal the wound and stop bleeding. So according to the specific stimulus, specific growth factors and cytokines, it gives us the specific cell. So again, stem cells are functionally undifferentiated cells that are capable to proliferate and have self-renewal to maintain the hematopoietic stem cell pool, give rise to a large number of differentiated progenitor cells. It maintains the population by dividing to undifferentiated cells. It is heterogeneous in respect to stimuli, as I mentioned before, according to the growth factor or growth hormone, to growth factor like thrombopoietin, erythropoietin, it can a granulocyte colony stimulating factor. It gives a specialized kind of cell. When they are proliferate, these hematopoietic stem cells, at least some of the daughter cells remain as hematopoietic stem cells. So the pool of the stem cell does not deplete. This phenomenon is called asymmetric division. These asymmetric divisions, that means that, I will show you here, when we have, this is the stem cells, when it's divided, it gives rise to daughter cells. These two daughter cells, one of them have the same characters of a stem cell and will not proliferate again and divide again. And the others proliferate and divide and give rise to different kind of terminally differentiated cells. This is the next figure. Stem cell divides into two cells. Okay. One keeps the capacity to have stem cells, and the other are proliferate, and after proliferation it differentiates into a specialized kind of cells and mature and go out to the peripheral circulation as a specialized cell. What are the morphology of these hematopoietic stem cells? Morphologically, it is resemble the small lymphocytes. I cannot distinguish between the stem cell and the small lymphocyte. But how we can know that this is the stem cell? We know the stem cells through cell membrane phenotype. By immunophenotyping, with a monoclonal antibody has identified them by the presence of a specific surface marker. So hematopoiesis, again, production and development of blood cells. Pluripotent stem cells are cells that have the ability to self-renewal and give rise to all blood cell compartment and all blood cell line. It needs growth factors that promote proliferation and differentiation, and then it mature to be released in the circulation as mature blood cell element, white blood cells, or RBCs or platelet. So each stem cells have the capacity to replicate, proliferate, and differentiate into specialized cells. They give rise to myeloid or lymphoid lineage. This myeloid lineage will give rise to and divides into monocytes, neutrophil or basophil or eosinophil or erythrocytes or megakaryocytic, or there is a lymphoid lineage which give us the T cell and B cell or natural killer cell. So all the blood cells are divided into three lineages: erythroid, lymphoid, and myeloid. Okay. Erythroid lineage, in which the end product are mature RBCs, mature erythrocytes. These mature erythrocytes have the function of oxygen carrying capacity, and the mature end product of this erythropoiesis is reticulocytes, which is immature RBCs that can present in the peripheral blood smear and in the peripheral circulation in the percentage of 0.5 to 2.5% of the total erythrocytes in the peripheral circulation. And the mature erythrocytes, which are difference in the concentration between male and female. The erythrocyte count is vary from male and female due to the androgenic effect of hormone. Male hormone stimulate erythropoiesis, and due to the menstrual loss of the blood loss during menstruation lead to decreased amount of erythroid bone. So the RBCs count in female almost range from 3.8 million per cubic millimeter to 4.8, while in male it is the range from 4.5 to up to 5.5 per cubic millimeter. Lymphocytes are the cornerstone of the adaptive immune response. They are derived from a common lymphoid progenitors. This lymphocyte lineage is primarily composed of T cell and B cells. This is during lymphopoiesis. We'll take it in separate lecture. Granulopoiesis or myelopoiesis is the formation of the granulocytes. These granulocytes are important in host defense mechanism and in adaptive immunity and innate immunity, and also this myelopoiesis give rise also to platelet and monocytes, which are important in coagulation process, and granulocyte important as a host defense mechanism. The term, you may hear this term, granulopoiesis. Granulopoiesis means the formation of granulocytes. Granulocytes are a form of white blood cells which have a granules in their cytoplasm. Okay. Megakaryopoiesis is the formation of platelet from the megakaryocytes, from the stem cells. Okay. How this happen? Okay. So we have here the multipotent hematopoietic stem cells, which have the ability of self-renewal and proliferation and differentiate into specialized cell element. Here I have multipotent stem cells. It can be proliferate and it can differentiate into other common myeloid progenitors or common lymphoid progenitors. This is upon what? Upon specific stimuli and upon it depend on the growth factors that is exposed to it. The common myeloid progenitors are give rise to either megakaryocytes and end with thrombocytes formation, which is the platelet, or peripheral unit and end with the erythrocyte or RBCs formation, or myelopoiesis, in which there is a myeloblast. These myeloblasts are further divides and it's either give rise to basophil or neutrophil or eosinophil or monocytes. Okay. The other lineage is called, this is stem cells, it can go to common lymphoid progenitor. These common lymphoid progenitors either give rise to natural killer cells or it gives rise to small lymphocytes where it either differentiate and takes the characters of T cell lymphocytes or it divides and proliferates into a P lymphocytes which end with a plasma cell which is responsible for antibody production. So, hematopoiesis, pluripotent hematopoietic stem cells are become committed stem cells and then it become differentiated. It gives rise to differentiate into a specialized cell type, then go released into the circulation. The bone marrow form a suitable environment for stem cell growth and development. The bone marrow composed of stromal cell and microvascular network. So, the regulation of hematopoiesis should be in balance, okay, to between cell production and cell death. Because if there is excess cell production, it can lead to cancer, and increased cell death, it can lead to aplasia, which is, both of them are dangerous situation. So, low regulation of hematopoiesis, it have an local environment